Welcome to our live class of genetics. Now, today we are going to concentrate on the mutation process. Okay, so before that, how was the yesterday's class? There was no comments from your side. Please let me want to say that let me have some feedback from your side so that it will be useful for me to improve myself. Is that clear? And welcome to our today's session. So, what is today's topic? Today's topic is mutation. Yeah. So, have you heard about the term mutation? Any answers? Don't know. So, mutation is nothing but a change. Okay, it can be a sudden change. It is an inheritable change in the chromosomes. Yes. So the genes know they automatically there will be changes taking place like a deletion process, like substitution process, where that substitution all these changes will takes place in the nucleotides. Yes, correct. So like adenine, we have seen no as during the structure of DNA molecule. I told you that the adenine, guanine, cytosine, thymine. So these are the nucleotides, that is nitrogenous bases which are present in the nucleotide. So when there is a change in this nucleotide only, that is the nitrogenous bases, certain change, there may be a deletion. For example, then adenine will always form a hydrogen bond with thymine, no? So instead of this thymine, what will happen when cytosine will come and occupy the place? It is a change only, no? Yeah. So like that, instead of the original nitrogenous base, the another one will come and occupy the space. So this is an example I am telling you. So like this changes will occur in the genetic sequence, in the chromosomal sequence. Yes. So that change is known as the mutation. Yeah. So it is the inheritable change, genetic change, we can say that. So this mutation is of two types, chromosomal and the genetic. Okay, so chromosomal means in the chromosome structure. So that is, chromosome means is the combination of many genes. Yeah, it's a combination of many genes. Genes are the combination of many DNAs. Yeah, so this chromosomal change, there will be a sudden change in its structure or number of chromosomes. It's known as the chromosomal mutation. And then this gene mutation is nothing but a point mutation, we can say. Gene mutation is also known as a point mutation, and this mutation can either be change the nucleotide sequence of a gene. Clear? It will be a changes in the nucleotide sequence of a particular gene. Understood? So there are two types of mutations, that's chromosomal mutations and the gene mutation, which is also known as the point mutation. Understood? So first of all, what I said, that mutation means what? It is the inheritable change. It is the inheritable change that takes place, okay, genetically, that takes place genetically. Understood? So this is about the mutation process. Have you heard about this mutation anywhere? What is the so who has discovered or introduced the term mutation? Any idea? This term mutation was introduced by the scientist who is called the Hugo de Vries. Okay, so he was the first who introduced the term mutation in genetics. And what he observed, suddenly he observed changes in the evening primrose plant. See, if we see that evening primrose plant, no, in this phenotypic changes were observed by him. So what is the phenotypic changes? Phenotypic changes are nothing but the physical triads. The appearance, the physical appearance changes were observed in this evening primrose plant by the scientist Hugo de Vries. So he was the person who found the term mutation by observing the phenotypic changes in these plants. Understood? So now, what is the definition of the mutation? Is an inheritable sudden change in the genetic material. That is, what is the genetic material? DNA. So we are concentrating only DNA in our class. So this mutation which takes place in the base of the cell, that is the base of the cell, is DNA. This DNA, when it undergoes any changes, that is known as the mutation. Understood? So this is a definition of the mutation process. Yeah. So an inheritable change in the genetic material, that is the DNA of an organism, is called the mutation. Understood? So if you have any doubts, please comment me, let me know that, by so that will be helpful for me to improve myself also. Okay. So next is, we will see about the chromosomal mutation. There are two types of mutation I told you just now. What are they? Chromosomal mutation and the gene, which is also known as the point mutation. Understood? So this chromosomal mutation, there are again of two types, that is changes in the structure of the chromosome and changes in the number of the chromosome. There are two types. One is the changes in the structure, another one is the changes in the number. Understood? So what is the structural changes, chromosomal structural changes? That's mainly due to the errors during the cell division. Okay, mainly due to the errors in the cell division. Understood? So what will happen? There may be deletion, there may be duplication. Just now I showed na, instead of thymine, any guanine or any other cytosine will come and place. No, it's a change. I told you that thymine and adenine only will form pair, will bond each other, and similarly cytosine and guanine only will form a hydrogen bond between them. This is the natural. This is the Chargaff's rule. Okay, there will be equal number of adenine and equal number of thymine, and similarly cytosine and guanine. Understood? In this, any mismatch, any deletion, any inversion, any substitution, okay, so all this will result in the mutation. That is change. Understood? So now we are seeing the chromosomal mutation in structural. Okay, so this is mainly due to the deletion process or duplication process. Deletion means completely it is removed. Duplication means one for the another one. Inversion, this is also a reversal. Inversion means reversal. Like what to say, mirror image, we will say, no? Mirror image. Your left right inversion. Have you studied in your lower classes? Like that, inversion also sometimes will takes place in the nucleotidic base, and then there will be translocation. Instead of one substance, another one will come and occupy, that is translocation. Understood? So this is a chromosomal mutation in structure. That's the first mutation. Understood? And the next one is chromosomal mutation in number. It may be either addition. There will be an addition of nucleotide base or a deletion of the number of chromosomes. So this is the change called the ploidy. That is chromosomal mutation in number, I said, no, that is known as the ploidy. And this ploidy, this chromosomal mutation in number, is further divided into two types, euploidy and aneuploidy. Okay, so this is about the general chromosomal mutation in the genetics. Clear? And let me explain about the ploidy. That is, ploidy means what? It is nothing but the mutation in the number of chromosomes. So there are two types of ploidy. They are the euploidy and the aneuploidy. Understood? So now we will see about what is the euploidy. Euploidy is nothing but the chromosomes would be more than the usual number. Okay, For example, I told you here some examples also. For example, instead of 2n, what is 2n? It is the diploid chromosome. This is the normal one. Instead of the diploid chromosome, if there is another one haploid is joining with this 2n, you will get 3n. Yes, so this is known as the triploidy or triploid, you can say that. So three haploid sets will be there in the chromosome structure, and then these plants and animals which have this 3n chromosomes, no, they will be sterile. What do you mean by sterile? Yeah, what is mean by sterile? You can come with your answers. Yeah, actually today I told you that I will conduct a small test, but after completing this mutation process we will have together a big test tomorrow with a question and answer session. Please be prepared for that, and I need those who are participating in this live session, I need your cooperation. I request you to comment, please. Okay, simply you are coming in and then going away. I want your comments, your what to say, your good comments, as well as any other negative feedback also I accept, please. So this is the sterile. This is what is sterile. There will not be any production. There will not be any production of plants. New plants will not be produced, and then the animals will not reproduce. Okay, that is known as the sterile. Understood? So now, like that, like 3n, there will be 4n also. 4n number, that is 4 haploid sets of chromosomes will be there in some plants. Here it is this 4 that is 4n chromosomes, no, it is good. There will be more production of plants, that is with the flower and fruits. Okay, there will be more fruits, and then flowers also. This is triploidy, that is, sorry, tetraploidy. Tetraploidy means it is the 4n, that is 4 haploid sets of chromosomes will be there. Understood? So that, like this, euploidy means that is more chromosomes than usual number. Okay, the chromosomes will be more than usual number. That is the euploidy. Understood? So this, in this we have seen two examples. One is the 3n set of chromosomes. If this 3n set of chromosomes, like a 3 haploid sets of chromosomes are there, mean they are sterile. And then if there is one more haploid set like 4n of chromosomes are there, means the plants of this kind will be having more fruits, and then they will be having the flower size will be also more. So this is one type of a chromosomal mutation, that is in the number of chromosomes. There is a change in the number of chromosomes. Is that clear? And the next is we are going to see about the aneuploidy. This aneuploidy is nothing but it is the loss or gain of one or more chromosomes. It may be either loss or the gain. Okay, that is addition or deletion. We can say. So here, what are they? 2 or 3 types will be there. What are they? Monosomy and the trisomy and the nullisomy. Monosomy means 2n minus 1. Trisomy means 2n plus 1. And then nullisomy means 2n minus 2. Nullisomy means 2n minus 2 is 0. So that is monosomy. If you subtract 1 from the that is diploid set of chromosome, you will get only one chromosome. If you subtract again, if you add 1, means it is trisomy. That is 2n will become 3n. Understood? See, look at here. I am going to subtract 1 from this. What will happen? You will get 2n minus 1, 1n. If you add 1 chromosome to this 2n, you will get 2 plus 1, 3. This is 2 minus 1. This is 2 plus 1. Okay, and this is trisomy. And nullisomy, you are going to subtract 2 minus 2, 0. So you will get 0. That is nullisomy. Clear? So this is about the aneuploidy. Aneuploidy is nothing but the loss or gain of one or more chromosomes in a set. It may be either the gain or it may be the loss. Clear? And the next is this aneuploidy, it is found in many conditions, many disease conditions. For example, I have given you the Down syndrome. So this Down syndrome, it is mainly due to the number of chromosomes in a set. Okay, so it is mainly due to the number of chromosomes. That is, for Down syndrome is one of the mutation that comes under the aneuploidy, and then it is mainly found by a doctor. Okay, initially it was found by a doctor Langdon, okay, in the year 1866. So here what is the defect? It means the chromosome 21, it will be the extra copy will be there, one extra copy, that is trisomy, like this, 2n plus 1. Instead of XX, one more X will be there. So this is the trisomy. Okay, so and the next is, if in this Down syndrome, okay, which is the aneuploidy, how that the individual will be mentally retarded. Okay, mental retardation will be there, and then their development will be very very delayed development, and then they will be having some behavioral problems if this aneuploidy is there, means that is the Down syndrome. And then they will be having weak muscle tone, vision and hearing disability. Okay, so these are the what to say, the symptoms or the clinical complications, we can say, when the individual is suffering from the Down syndrome, which comes under the aneuploidy mutation. Aneuploidy is nothing but the loss or gain of one or more chromosomes in a set. Okay, so what will happen here? This gain actually, yes or no? So here there is a gain. So that the extra copy of chromosome 21, instead of diploid, usually all the chromosomes will be diploid only. Okay, so instead of this diploid, one chromosomes is added, means it is the mutation. So here the 21 chromosome will be having extra chromosome, that is known as the trisomy 21. Is that clear? And the next is, what are all the clinical complications they will be having? They will be mentally retarded, they will be delayed development will be there, and then the behavioral problems will be there, and they will be having weak muscle tone, vision and hearing disability. Clear? So these are the general symptoms of the Down syndrome, which comes under the aneuploidy. Aneuploidy comes under the chromosomal number mutation, changes in the number of chromosomes mutation. Is that clear? So far we have studied about the changes in the structure of chromosomes and then changes in the number of chromosomes. Now let me explain about the gene or the point mutation. This is another type I explained to you in the beginning during the introduction part. So now gene or point mutation is nothing but the changes occurring in the nucleotide sequence of a gene. This I told you in the beginning. So what will happen? The nucleotide sequence of a gene, that is the point mutation or the gene mutation, we can say. Okay, so what will happen here? Here there will be the nucleotide substitution will be there, nucleotide deletion will be there, nucleotide insertion or inversion of a single or more than one nucleotide base. Understood? So for example, I am giving you now okay, and the next is C is for joining. Okay, here instead of this and this, what will happen? Again A is coming, instead of thymine A is coming, means it is a mutation. Here instead of G is C is coming again, it is a mutation. Yeah, it's a point mutation. A particular nitrogenous base is changed. Clear? So this is the mutation or gene or point mutation. Understood? So if there is a gene or point mutation is there, means what will happen? The gene which is responsible for the protein formation will be abnormal. Yeah, so you will be getting abnormal protein. You will be getting abnormal protein. I told you in the previous class that DNA is responsible for RNA formation, and then RNA will produce the protein. Okay, So now if there is a mutation in the DNA itself, what will happen? That muted gene only will form the RNA, and again that RNA, muted RNA only will form the protein. So this protein will be abnormal. It will not be a normal protein. Clear? So about the gene or point mutation. Okay, so if you have your friends or relatives among your friend circle or family circle, you can share my videos, and then you can to say that subscribe to my channel. I am taking live classes for the what to say that life science students, and also the biology students, and then chemistry. Okay, class 10 and class 11 and 12 biology, biochemistry, then class 10 science. Okay, on all the life sciences. Please share among your friend circle so that more people will watch my video. Okay, so today what are the things we have studied in our class so far? I started the session with the mutation. Okay, now it's the question. Shall I pose some question? It's a question session. Who introduced the term mutation? Who introduced the term mutation? It's nothing but the Hugo de Vries. And how did he find that? He found phenotypic changes in the primrose plant. Okay, so that he came to a conclusion there is a sudden change in the gene. Okay, so he found that what is the mutation? It is an inheritable sudden change in the genetic material, that is the DNA. In the DNA there is an inheritable change. Inheritable means what? It is by birth itself, it is there is a change in it, in the gene combination itself. Okay, it's not acquired, it is inherited. So by birth, in the gene itself, this DNA mutation has occurred, changes has occurred. This is the definition of mutation. Okay, so now if people are interested, you can join my online paid coaching. Okay, you can join my online paid coaching. 1-to-1 will be there. So I have given my email ID in the last session, so that you can contact me through the email. Okay, now next is what we have studied, mutation. We have studied about the mutation, chromosomal mutation and the gene mutation. There are two types, and this chromosomal mutation is nothing but the changes in the structure or number of chromosomes. Yeah, sudden change will be there. And the next is it is the gene or point mutation, we can say, is changes in the nucleotide sequence of a gene. Is changes in the nucleotide sequence of a gene. Okay, and the next is we have studied about the chromosomal mutation. Okay, so this chromosomal mutation in structure is mainly due to the error in the cell division happens. What are the errors? It may be deletion, it may be duplication, inversion, or translocation. Anything can happen in the nucleotide. Okay, and the next is we have studied about the chromosomal mutation in number. There may be an addition or deletion of the chromosomes. Understood? So there are two types. So this condition is known as ploidy, and in this ploidy there are two types, euploidy and the aneuploidy. And in the euploidy, what will happen? There will be that is normally chromosome will be present. Along with that, one more haploid set will be there. That is would be usual. It is unusual more than the usual number. Okay, so next is for that we have seen about the examples. Triploidy, 3n. There will be 3 haploid set. If this condition is there, means the plant or animal will be sterile. Okay, they will not reproduce. And the next is tetraploidy. Here there will be 4 haploid sets. The 4n will be there. Here the plants of this kind, no, they will result in good fruits and the flowers production. Okay, so this is about the euploidy. And then we have studied about the aneuploidy. Aneuploidy is nothing but the loss or gain of one or more chromosome in a set. Yeah, it may be the addition or it may be the loss. So there are two types. If it is loss means monosomy, the diploid set, no, if it loses one chromosome it will become monosomy. If there is additional one is there means it is the trisomy. It is again it's completely lost means it is nullisomy. So there are three types: mono, tri, and the nulli. And this kind of aneuploidy will be found in many disease conditions. I have given one example here. It is the Down syndrome. Okay, it's in the Down syndrome. It is an aneuploid condition, and it was found by the Dr. Langdon Down in the year 1866. That's why it's called the Down syndrome. Since it was found by the Dr. Down, it is the Down syndrome. Okay, here what is the condition? The 21st chromosome will be one extra copy, that is known as the trisomy. The condition of Down syndrome is trisomy. It is having one extra copy. And then the children will be mentally retarded. Those who are with this Down syndrome, additional chromosome 21, they will be mentally retarded. Their development will be delayed. They will be having some behavioral problems there, and then muscle tone will be very weak, and then their vision and hearing also very weak. They will be having disability in the vision as well as the hearing. Understood? So hope you would have understood today's session. Yeah, about mutation. My explanation would have cleared all your doubts, I think. Okay, now what is the gene mutation? I told you that gene mutation is mainly due to the nucleotide sequence of a gene. Okay, what are the conditions here? Deletion, substitution, insertion, inversion will be there. I already explained you about this adenine when it should be paired with the thymine only. If it is not pairing, any other adenine will coming and occupying, it is a mutation, it is a change. That change will be producing abnormal protein. So one gene alteration is there means it will result in the abnormal protein of an organism. Understood? So this is about the mutation. Okay, clear? So tomorrow we will be having a test session. That is it may be a discussion also. I will give the question. You can come out with your answers, or I will give the answers to you. Okay. Next is those who are willing to come and join my online paid coaching. You can join. This is my email ID. You can contact me through this email. So I am here to place classes for the class 10 science, and then 11 and 12 chemistry and biochemistry students, and then class 11 and 12 biology students, and class 11 and 12 physics also. Okay, in turn general life sciences, life science students also can join. If you have any doubts, you can also join, like biochemistry students, and then biotechnology students. Okay, you can join for the online course. It is a paid coaching only. Okay, but it will be one-to-one interaction will be there. Okay, and I can assure you that the class will be very useful for you if you join the coaching. Okay, thank you. We'll see you in the next live session with the test session. Understood? Okay, bye. Take care.